Welcome back. In this particular video, we're going to talk about visual sensation. So primarily in this video, we'll focus on how we capture information in our eyes, visual information. For that, we need to know the actual stimulus. So we'll talk a little bit about the light, the stimulus itself, and the primary focus will be on the structure of the eye and how it captures the light information. Light is both particulate in nature and it also has waveform. And for vision, the wave nature of light is very important, and that's what we'll talk about. Light as a wave is an electromagnetic wave. It has both electric and magnetic waves in it. And this electromagnetic wave, whenever you have wave, you have wavelength, right? And this electromagnetic radiation, which is in the waveform, is the important stimulus for us to see. Right. So the electromagnetic radiation itself, the spectrum is has different wavelengths, meaning different kinds of waves, starting from, for example, the radio waves, the microwaves, the infrared rays, the ultraviolet rays, X-rays, gamma rays, and cosmic rays, and whatnot. But we only perceive only a part of this electromagnetic spectrum, which we call the visible light. And this visible light itself has individual wavelength components, which gives us the colors specific to the rainbow. So this is just one visible aspect of the electromagnetic radiation that we can perceive, but there is a lot more than what we can perceive. So the visible spectrum, which has specific wavelength, is often the white light, but it further is divided into its individual components. And you might have studied this in your middle school or high school as GC Roy, or the way I learned is VIBGR, VIB GR, GYOR, violet, indigo, blue, green, yellow, orange, and red, in the increasing order of the wavelength, meaning that the violet has the least, the shortest wavelength, and red has the largest wavelength. And this is the only part of the light or the electromagnetic radiation that we can perceive. So that you can see when the white light goes through a prism. It's called prism effect. You can see that it breaks down into its individual components called VIBGR, or however you want to call it, violet, indigo, blue, green, yellow, orange, and red. Now that we talked about the light itself, I would like to say that the next thing to talk about is the eye. There's one more specific thing I want to tell you about the light itself, is that the part that we can only perceive the visible spectrum of the light, and the fact that it has individual components related to the 7 colors or the combinations of those, is the reason why we can perceive the world in colors. So depending on what color is reflected from an object, we perceive that color. For instance, if you're looking at a red car, which is right in front of me, in front of the window, I'm right behind the window where I am sitting right now, the red light is reflected from that object, and that's the reason why I can see that as a red car. If it's a blue light that gets reflected from that object, I'll see that as a blue car. So the idea is that just because the light that gets reflected from the object, we perceive that object in that color. Now, the specifics of how we determine the color itself, we'll talk about it in the color perception video, but in the rest of the video, we'll primarily focus on how does eye capture this light information and how it gets sent to the brain where it eventually gets perceived. Okay, coming to the structure of the eye. This is the front view and this is a side view. And we'll talk about the details of the side view of the eye in the next slide. For here, I just wanted to give you the basic elements of the eye structure. If you look at it, it's a circular structure. It's like a ball. That's why we have eyeball, right? And with a narrow opening. This narrow opening is covered by, right, this circular muscle. You see that? This is a circular muscle. This circular muscle that covers the opening of the eye is called the iris. It is that structure that gives color to our eyes comes in different colors like brown, hazel, blue, and whatnot. Right behind this iris is this structure called the lens, and we'll talk more about this in the next slide. So the narrow opening that is left here, right, because of the circular muscles, which can determine the size of this opening called pupil, right? It can contract or relax, and thereby adjusting the amount of light that can enter your eye. So these are the basic structures. The iris, the opening, behind that there is a lens, and behind the lens there is this eyeball itself filled with fluid and whatnot, and there are muscles around the eyeball that controls the movements of the eye. And we'll talk about more structures in the next slide. Okay, coming to the structure of the eye, as you can see, the eyeball itself is like a bowl or a pot with a narrow opening. And this narrow opening is covered by this muscle, the circular muscle called iris. And it can contract and relax because it's a circular muscle. When it contracts, this opening left by the iris called pupil, right, it decreases in size. And when it relaxes, this opening will increase in size. So when there is more light, the iris will contract so that only limited amount of light can enter your eye. And when it relaxes, especially when you are in a dark room, it relaxes so that the opening, this area, increases and more light can enter your eye. Now, the first structure that's behind this iris is this lens. And light passes through this structure, a protective layer called cornea, then goes through this opening and it passes through the lens. And because of this lights get reflected onto the innermost layer, the inner layer of the eyeball. And this inner layer of the eyeball is called the retina. Okay. So again, the cornea, the opening left by the iris is the pupil, and right behind is the actual lens, and the light that gets refracted by this goes and forms an image on the retina itself. The retina is a continuous layer of cells, and there are multiple layers within this retina, and we'll talk more about it later on. And there is a particular spot inside the eyeball where there is no retinal cell, and we know the details of it. We'll talk about the details of it in the upcoming slides. But there is one spot in the eyeball where there is no retina, right? This is the exit spot for the axons to form the optic nerve. So from each eye, optic nerves come out of the eyeball, and the blue things are not the nerves. These are the blood supply to the retinal cells. The exit is called the optic disc or blind spot. But there is also one particular space, one part of the retina, which is kind of the center of the retina. It's called fovea. This has more of a specific kind of photoreceptors called cones. We'll talk more about it later on. And the light falls on this part of the retina called fovea. You can see the details. When you focus on an object, its image is formed here and you can see the details of it. But if the image is formed here, the exit point for the nerve, the blind spot or the optic disc, right, you can't see because there is no retinal layer here. Okay. So then the optic nerves from each eye, right, they eventually go to the occipital cortex, which is the visual center, what its function is. So when you're looking at an object, right, the light reflected from that object goes through the cornea and the pupil, and it goes through this particular structure called the lens. Now here's the thing, the lens can adjust itself so that this image that you're looking at, or the object that you're looking at, the light reflected from this object that you're looking at, falls on the retina. And this process is called accommodation. You can see the object clearly only if the light reflected from this one falls on the retina. It's just like your projector projecting onto the screen. If the image is projected behind the screen or in front of the screen, you won't be able to see it. It has to fall on the screen. Just like that, the light reflected from this object should fall exactly on the retina. If it doesn't, then you have difficulty in vision. And one such difficulty is called nearsightedness, where the structure of the eyeball is such that, you can see that, right? It's not circular and it's prolongated horizontally for various reasons, right? Then the light that is reflected from this object, right, it falls right in front of the actual retina in this person. And for this person to actually clearly look at the object, right, they go closer to the object so that the image now forms on the retina. This is nearsightedness. And most of the young individuals who wear glasses have this issue. Like I myself had this when I was in high school. Until then, I've been using glasses. What these glasses do is it corrects this lens. It's giving the extra lens, right? So that the image falls on the retina. So this deformity because of the difference in deformity in the shape of the eye, where the image is formed in front of the retina. It's called nearsightedness. Nearsighted individuals, they have difficulty reading stuff that's away from them. So if you're sitting in the last bench, you would come to the front row to actually read what it is, or go closer to the blackboard in order to see what it is. So that's nearsightedness. Nearsightedness is when you can only read things better when you're closer to object. And the opposite is farsightedness, and that's what we'll talk about in the next slide. So in farsightedness, which opposite to nearsightedness, right, you can see things clearly when they're far away. It's because of this deformity. In one of the reasons is the deformity in the structure of the eyeball, and it's not circular, but it is vertically oval shaped, right? And the light that is reflected from the object that you're trying to focus on, it's actually not from the retina, but it forms behind the retina. So the focal point falls behind the retina. So for the individual to actually clearly see, then they move their head back or move the object back, like the elderly. Like right now, like when I want to see something, if it's close, I can't see it. I try to move the book away from me so clearly see what it is. So that's farsightedness. Farsightedness is when you can clearly see the object when it is far away. When it's close, you can't see it. Well, at certain age, it's a combination. That's why people wear bifocal glasses, right? Where they have both farsightedness and nearsightedness. So the lenses that individuals with farsightedness wear would help this particular lens so that it's giving that extra lens, right? And the image will eventually be formed right on the retina as opposed to behind. Okay, so in the next slide, we'll look at the detailed nature of the retina itself, right? So what we'll do is we'll take this part of the retina, right, and zoom in and look at the different cell layers and the optic disc. And so in the next slide, we'll see a zoomed in version of this part of the retina. Okay, so let's look at the retina itself. The retina it's multi-layered, and these are the most important cell layers. The first innermost layer that you see here is the layer of photoreceptors, rods and cones. You can see the color blue and red. These are the photoreceptors that are sensitive to light. These are the actual neurons that capture the light energy and convert them into neural impulses, which then goes to the next cell layers and eventually comes out of the eye through optic nerve. The second important layer of cells is the bipolar cell layer. Optic, the photoreceptors, bipolar cell layers, right? And the last layer is the ganglionic cell layer. So ganglionic cells, bipolar cells, and photoreceptors. There are other cell structures in between, but we'll only focus on these three types of cells: photoreceptors, bipolar cells, and the ganglionic cells. Right? As you can see, the photoreceptors are connected to bipolar cell layer, which are again in turn connected to the ganglionic cell layers. And the axons of these ganglionic cell layers, which is the last layer of the outermost layer of the retina, right? The axons of it, right? They come out of this exit. Remember, we said there is one particular part of the retina where there is no retinal eyeball, where there is no retinal cells. Well, that's because the axons of all these ganglionic layers, they accumulate and form a bundle of axons. This bundle of axons of the ganglionic cells, right, that come out through this exit called optic disc or blind spot, right, as optic nerve. Okay, so that's why when light falls on the optic disc, right, there are no retinal cells to actually capture the light. So this is what happens when light falls on the retina. It goes through, cornea, pupil, lens, and the light falls on the retina, right? The light actually physically passes through the cell layers first and reaches the photoreceptors. These photoreceptors then convert the light energy into electrical impulses, the neural impulses or the action potential. These neural impulses or action potential is then transmitted from photoreceptor layer to bipolar cell layers, from bipolar cell layer to ganglionic cells. From the ganglionic cells, through the axons of it, they exit the eye and then they reach the uripe, the cerebral cortex and the occipital lobe. And in the next few slides, we'll talk more about the photoreceptors, their properties, and their importance. Okay, so talking about photoreceptors, right? And we said that the bipolar cells and the ganglionic cells. So these are photoreceptors. There are two kinds of photoreceptors, rods and cones. These are rods and these are cones. Okay, and we'll talk about this, the nature of these photoreceptors in the next slide. I just wanted to give you an idea. Remember we talked about the fovea earlier, right? The center focus. This fovea is filled with colors. Cones. By the way, the cones are meant for color vision and rods are meant for dim light vision. And we'll talk more about it in the next slide. Okay, so the rods and cones. There are about 120 million rods and only 6 million cones. Rods are the photoreceptors, meaning the specialized neurons that are sensitive to the brightness, right? And they are not sensitive to the color, and they are more active or useful in dim light. So they are meant for dim light vision. And there are more of these in the periphery than at the center. So that's why they're responsible for the peripheral vision. What I mean by that is when you look at the structure of the eye, right? Let me draw this vague picture. We said there is a fovea here, right, at the center. Now, if you three-dimensionally imagine it, right, in the fovea, there are 100% of cones, right? And as we move away from the fovea, right, to the periphery, right, then the number of cones decrease and rods increase, so that lots of rods at the periphery and no rods at the center. So that's why when there is an object moving in your peripheral vision, you can identify that there is an object that moved, but you cannot determine the details of that. For you to determine the details of an object that's moved in your periphery, right? You're reading a book and somebody moves in your peripheral vision and you turn your head and say, who is that, right? You can't say who that is. You can just say that some object moved in your periphery, right? That's because there are more rods in general, but there are more rods in the periphery than at the center. Then coming to cones, they're sensitive to light and they're only 6 million and they're less than rods, as you can see. They are for daylight vision and they're responsible for determining the fine details in the bright light, and they're more at the center. So that's why when you're looking at an object and trying to figure out to know the specifics of it, right, you put it close enough that the image of this object falls on your fovea, which is 100% cone cells, which are responsible for capturing the fine details of light, including the color. Okay, so what happens, right? Well, when light passes through the cornea, pupil, then through the lens, then it's captured by the retinal cells, right? And go from the photoreceptors to next layer, bipolar cells to ganglionic cells, and the axons of the ganglionic cells, right? And the information exits the eye through this bundle of axons called the optic nerve. Then eventually it goes to the brain. Now let's see the last structure involved in vision, namely the brain. So the light energy that is captured by the photoreceptors eventually comes out of each eye through optic nerve, and the optic nerves go from each eye, and there is crisscrossing of fibers. We'll talk about that in the next slide. And eventually it goes to the thalamus, the relay station. From there, the information goes to, or the fibers reach the occipital cortex. So this is the direction of flow of information from the eyes through the optic nerve to, you're right, thalamus, and from thalamus to, you're right, cerebral cortex and the occipital lobe. Okay, so in this slide, we'll talk about few constructs: contralaterality and the contralaterality in visual system, and how that is determined by the visual pathways. Contralaterality, contra, opposite, lateral, the side, right? Usually our system is contralateral, meaning that the sensory information from, say, left side of your body is sent to the right hemisphere, or the right side of the body, or the left side of the body, for instance, is controlled by the right hemisphere. So this is contralateral. One side of the body is sensed by the opposite side of the cerebral cortex. And this is called contralaterality. Contralateral. So for instance, take an example. Somebody touches your left hand. Now you can perceive the touch because the information from your left hand eventually goes to your right cerebral hemisphere. Particularly, it will go to the parietal cerebral cortex. Remember the sensory cortex in the parietal lobe? It goes to the right side. If somebody touches your right hand, those sensations, those electrical impulses are sent to the left hemisphere's sensory cortex. So one set of the body is sensed by the opposite hemisphere. Okay, so that's called contralaterality. With the vision, though, it is slightly different. You cannot say that the information captured by left eye goes to the right occipital lobe and vice versa. You cannot say that. In fact, information going from both left and right hemisphere, sorry, left and right eye, right, information goes through both and goes to the opposite hemisphere. This is like contralaterality at a different level. Okay, let's talk about that. If you look at it, the optic nerve fibers, right, they crisscross, but they crisscross in a very unique fashion. See that? Let's look at the left eye. The optic nerve from the left eye, right here, right, half of them, especially the medial half, the ones that are centered to the center, the medial half, they go to the opposite side, but the lateral half go to the same side, right? And the same here. So there is crisscrossing of optic fibers or optic nerve fibers, but only half of them, right? This leads to a weird situation where you cannot say that the light captured by left eye goes to right hemisphere. You can't say that because it goes both to the right hemisphere and also left hemisphere. But here's a weird thing, or the interesting thing is that it is contralateral at the level of the visual field. Well, what is visual field? Well, if you look straight ahead, right, whatever kind of you can see is your visual field. And if you divide that visual field into left half and right hand, and let's assume for our conversation here, the left side is red in color. So you see the red part, right? Red. And the right side is blue in color. So the right of your hemisphere, sorry, visual field, you're looking ahead and all the right side is blue in color, left is red in color. And as you can see, information that is red on the left side, it passes both through the left eye and also the right eye. And because of this crisscross nature, eventually it goes only to the right hemisphere. Information in the left visual field goes to the right hemisphere. Similarly, the information that is blue on your right visual field, right, that passes through both these eyes and eventually it goes to the left hemisphere. So information in the left visual field goes to the right hemisphere and information in the right visual field goes to the left hemisphere. So there is contralaterality, but at the level of the visual field, not at the level of the eye. And this peculiar, unique crisscrossing of optic fibers from the opposite eyes, from these two left and right eyes, results in this X-shaped structure, right? And this X-shaped structure, this crisscrossing is called optic chiasm. Optic chiasm. Okay, we've just seen that the information captured by both the eyes, right, through optic nerves reaches, it goes through relay station thalamus and eventually it goes to the occipital lobe, cerebral cortex in the occipital lobe called visual cortex. Now, Hubel and Wiesel in 1981, they received Nobel Prize for their discovery of the specialized cells in the visual cortex in the occipital lobe, and they call them feature detectors. And we'll talk about these feature detectors in the next slide. So what these researchers did is they placed an electrode, a mini electrode in the visual cortex of this cat, and these electrodes can capture the firing of specific nerve cells at the level of the neuron. And they say, okay, this particular nerve is active. Well, this particular nerve is active. What they did is they made this cat look at some images. And what they found was that there were very predetermined specific cells in the cerebral cortex, the visual cortex, in the occipital lobe of the cat that only fired when they saw horizontal lines, right? Or only, and there were other cells that only fired when there were vertical lines, and there were some neurons that only fired when when the image was of that of angles. So the idea here is that our visual system works in a way that our cerebral cortex have specific receptors, or sorry, specific neurons that are sensitive to specific features in the object. In other words, what you're doing is that your brain is kind of dissecting an image into its basic features. right? And these basic feature cells are the ones that fire. Now the question is like, well, when you're looking at something and if you're breaking that into individual features, how do you combine these things? Well, according to the research, there's a lot to know, but according to research, we actually combine these features again at the occipital level to actually make sense of this and get a bigger picture of what this is. And these particular neurons that are sensitive to specific features in the object are called feature detectors.